Dr. Ghosh, Madam Ghosh, Dr. Mehta, Dr. Krishnamurthy, scientists of this institute, both retired and present, invited guests, ladies and gentlemen. Probably this dais is nothing new to me because I have been here many a times, and not only to talk to you but also to organize many of the seminars and workshops. If I run through the slides to tell you how this small thing took place in this institute and went on and culminated into a discipline, you will be a bit surprised to know about this. I joined this institute in the year 1961 on 26th June when Dr. Iyenger was the director. I had the interview on the 24th of April. He asked me, "You come and join now itself." I told him, "Sir, I am already working somewhere else. I have to hand over charge and come. You write from here itself, that charge. Leave it there and send it by post. And start your work today itself." Then I told him it's not possible because I have to hand over physically everything, stores, accounts, everything I have to do. So later on, I begged excuses and then went back, handed over charge, came here and joined on 26th June. Dr. Kappana called me and said, "Come on, you take up this project on chlorella." I thought, what is it? I have come here to do this work on chlorella. No. First you do this work. Let me see how you succeed. Afterwards I will do some other work. Then I started the work. When I was doing it in the laboratory, he said, "Oh, it's not sufficient. You go and do it on the terrace. We have no place here to put up ponds and all that. You go to the terrace, and when you get the ponds built, you start working on that." Then I started. Every day we used to go in the summer and sit there. It used to be very hot. Put a skull cap and sit there and start my work. Fortunately for us, we could isolate a very excellent strain of chlorella, and we could do good work and make that a landmark in the chlorella mass culture in India, which was done for the first time in India itself. After that, one day he called me and he said, "Look here, you have completed this work. I have got another work to do." I said, "What, sir?" I thought he's going to give me something which I can't do. He said, "Yes. You take seawater and put it on crops." I said, "Sir, it will die. Let it die. Why do you worry? You first do it." I said, "Okay." Then I brought some pots, put sand in that, and then put some seeds and grew it, and then started pouring seawater. Everything died. Then I got disheartened. What can I do? This man says, "You do it. What will be my career? I am only 26 years old. I have to do some more work and get on in the research field." Then what happened was, he gave me a reprint of Dr. Boyko and said, "You read this, plan your work. I am going to CSIR. The scheme has not been approved. After that, you can start working on it." Then I sat for 2-3 nights, went the midnight oil, and then prepared a nice plan and took it to him. He opened his cupboard, took some paper, said, "Say, whatever I have planned, you have planned, seems to be more or less similar. Go ahead," he said. That's how it started as a scheme in CSMCRI. And we started the work with local varieties And somehow when I was working on marine algae, my first work was on marine algae, we were doing the survey work. At that time I had noticed in the estuarine region, the enteromorpha was growing both in freshwater as well as in seawater and also in the diluted seawater. This occurred to me, why should we not also train our plants to take seawater later? When the nature is doing that, we should also try to attempt and do something. Then I started building up the salinity resistance in the plant by year after year. And later on we were able to grow bajra and wheat in the coastal region. This is a picture. Wherever you have the sea, the inseparable sand is always there, which cannot be used for any other purpose other than just growing some shrubs. Sea and sand are always inseparable. And we thought that we can get this seawater and then start using it. But the seawater salinity doesn't remain constant. It also goes on changing, right from the river dilution that takes place in the estuarine regions, and then it goes on increasing in salinity. If you train the plants to take up from a lower salinity to higher salinity, you should be able to grow the plants with seawater. That's how we did the work. Next slide. Where is the work? Next. Okay. This is how we started in the pots, because we should have a particular type of column of sand, otherwise the salt will accumulate at the root zone. It doesn't go to the deeper layers. Always an equilibrium of salt should be present in the root zone so that the root shock doesn't take place to the plants. After doing this pot work experiment, then we went to the field experiments where at the Hatab region, unfortunately the water is very muddy there. We cannot use the water straight away. So I thought of a plan. It is better to have a sump. Put a filter bed right from the sea shore to the our place of work, and then get a pure filtered water for our use. Amazingly, we found that the salinity did not rise very quickly. It was very slowly it started rising, and the plants were able to withstand that salinity, and we could get quite a good yield. Then I struck a deal with this man, because we could not go every day and do it. He was a farmer there. I called him. You do the irrigation and other things. We will give you money for purchasing, we will give you fertilizer and everything. Whatever yield we get, you take half and we will take half. He said, okay. And for nearly 3 years, first year he did, and then he said it is okay. Second year also he said, all right. Third year said, no, I don't want to do it. Why? I said, you take me here as a casual labor, give me a permanent job. I said, no, I can't do that. Then we again started other type of work in the laboratory, and then what I want to say is this is only a sort of a future plan. It is not the present plan for the people to adopt this immediately, because it needs a lot of research work to go in. You have got to select plants which are salt tolerant and then genetically modify it so that the plants can take up the salt without any problem in it, so that the yield doesn't get reduced and people will be able to use the technology. Next one. We attempted with a number of crops, not only one. We had sugar beet. You will be surprised that people said sugar beet is very good. We had a lot of discussions in the RC meeting. People from the ICR said, no, you take up something with plants which can tolerate very high salinity. So you should take up such a crop which will take up a lot of salt. And then we started with sugar beet. Oh, we could grow it because we could get a lot of yield and all that. But unfortunately what happened is all this salt started accumulating into the tubers. When we wanted to take out the sugar, it was not possible because the amount of salt present was more than the sugar. And if you want to remove it either by ion exchange or any other process, it will be very expensive and also not a feasible process. So what I did is, I simply chopped off all these things, got some this exact from CFTR, fermented it, made it into alcohol, and used that alcohol for running the knapsack sprayer. So I thought it can go as a fuel tomorrow. If it is not possible for using it as a sugar crop, it can be used as a fuel crop. So this was one thing which we did, and we thought that we should focus our attention on some other crops also. Like that we had a number of crops, something like 15 or 20 types of crops we had. Before I started this work, when Dr. Kapana told me you take it, I went to Indian Agriculture Research Institute, Delhi. At that time, Dr. B.P. Paul was the director of the ICR was not formed at that time. He was the director of the IRI. So I waited at his residence for about nearly half an hour. And he came out and he said, okay, what do you want? Gentlemen, why are you standing here? Sir, I want to discuss with you something. Yes, tell me what it is. I explained to him. It's all not possible. Why do you waste your time? You go, he said. Then I got disheartened. I went to the plant physiology genetics division. Dr. Swaminathan was doing some crossing work with the Mexican dwarfs with Indian varieties of wheat. He said, you wait here. Do you know how to do the crossing? I said, I know, sir. Come with me. I went into the field. We did some crosses. And then we came back. What do you want to say? I said, sir, this is what I want to do. It's a very difficult project. Why do you want to take this project? No, sir, this project I have not taken. It has been given to me. Now I have to work on it. He said, it's a very good project. If you succeed, you will make a name in the project. If you don't succeed, you will have to find out some other job. So it is up to you, he said. And I came back after, say, a few years. The ICR advertised a post of plant physiologist in the Central Soil Salinity Research Institute. And I applied for it and I was selected. Then I went there. When I went there, I broadened my horizon. How are we to deal with this agricultural problem? Because CSIR doesn't provide you this opportunity. When I went there, I found that you have got to work on it and then pass it on to the field staff to do the field work. And then later on, when you cross these things, you have to give it to the varietal trials. And then only you can put it, give it to the farmers. So there is a stage you have got to cross. You cannot simply take it and give it as we are doing in CSIR. Next please. You can see here, these are all the plants which we have taken up in cereals. We have taken wheat, a ball wheat we have taken which is known as Karchia. Amazingly, this wheat has got tremendous potential. It can take nearly 3/4 of the concentration of seawater right away and can grow right from seed to seed. It has got that capacity. And similarly, we did it with bajra, barley and all that. Our barley paper was referred by the Davis, the plant nutrition division of Davis, when they started working on tomatoes with saline water. I was invited to present these findings at the international conference which was held in Kiowa Island in North Carolina, South Carolina. And the professor of the plant nutrition department came to me and said, what is this you are doing? I told him, this is what I have done. I have already shown you this. No. This is not the way to do. You first of all find out wherever the plants are growing on the sea coast, take those plants and then the cultivated plants, cross these two things and produce a variety which can grow better with seawater. I told him, Professor Epstein, it will not grow. First year you will be able to grow it. Second year we will put this seed. Neither this is he said, I will take a tomato which is on the sea coast, which are smaller in size, and then I will take a well-developed variety of tomato and cross it. I said, you get neither. You will get middle-sized tomato, which will be useless, I said. Same thing happened. In 1980, he invited me and said, you please give a lecture to my graduate and postgraduate students about the work that you have done. I will tell my experience afterwards. When I started explaining everything, he said, look here, first I thought he is doing something which is not correct. But later on I found that what he has done is correct, what I did is wrong. So, what I want to tell is, we are in such a comfortable position where we have the sea, we have the sand, but this is only for the future. We have got to still develop and make this a profitable proposition for a farmer to take it up, because farmer will not take any technology which is bit expensive. Second thing is which is cumbersome. Third is which is not giving him any profit. If it is so, he will not take. So we thought that we can keep this in the background till we employ some people who can do the biotechnology and then genetic engineering on this, and then put it up later on in the field. Next please. Then we thought, why not we pitch upon some plants which are already growing in the saline land and then do some work on this. So, next. Then during my visit to Israel, and I didn't, this one, not Israel, to Egypt, I met one scientist, one Zahran. He was struggling to do some work with salt water and all this. He had a kind of grass called as juncus. Amazingly, this juncus had nearly 80 to 90% alpha cellulose in it, which could easily go in for making quality paper. Then I thought, why not to get some seeds from this man? I asked him, can you please give me some seeds? He said, no, I cannot give, because when you take it, quarantine people will catch you. You do one thing, you put this in a cover, post it to me, so that I can receive it there, and I will use it for my work. Same thing he did, he sent it a few seeds in cover, and then we started growing this. Amazingly, this plant has got a capacity to withstand even direct seawater, and it can grow. The clumps have got very good alpha cellulose in it, to the extent of about nearly 85 to 90%, and we could make very good paper out of it. We went to IICT where Mr. Jetkar was there. With him we had a project, and we made a beautiful paper. When I showed it to Professor Nurul Hassan, he said, no, can I write on this? It will soak? He said, you can write and see, sir. I said, he wrote it. This paper is good. Ink does not soak in it. He wrote and gave me. And he said, you continue this project, it's a very good project. And later on, when Professor Naidama visited our institute, then this pilu is one which is already growing on the saline land. We thought, why not we do the basic work in this, so that we can find out the high yielding plants in this and having high oil content, and also the composition of the oil doesn't change very much with the edaphic factors. So we pitched up on this and got both Salvador of Persica and Salvador of Oleoides, and we have got very good data on this. If somebody would like to use it, it will be an excellent thing to pursue further. Next please. We took it up in Kutch region where If you dig, you will get nothing but only seawater. So, one entrepreneur, he said, I would like to use my land for growing something. What would you suggest? I said, Pilo, अरे यार, क्या करेंगे उसको? क्यों तो बहुत, it will take long time. I said, no, I will see that it will give you some fruits within 2 years so that you can sell it in the market. And we got him some bank loan also. We certified that the land is good for growing this type of plant. And the Dina Bank gave him about 12 lakhs of rupees for him to start this work. When we were doing this work, night time the Pakistani fellows used to come on the camel to see what is going on here. And similar thing was translated in the Karachi area, where they used to grow this salvadora on the other side of the creek. I don't know whether further what they did, but anyway, this plantation is still existing in Kutch, and this man is now harvesting and then sending it to Aegis Industries for extracting oil and using it for sodium laurate extraction. Next, please. Then I contacted an Israeli scientist and said, I want to understand the mechanism of salt tolerance in many of these halophytes which are growing. Then I found that Atriplex was one thing which was very interesting to us. We got some seeds from Israel and then we got this put up in our Hatab area. When we irrigated with seawater, we found that the salts started were thrown out on the leaves. We thought, what is happening here? We found that the salt moves and then into these pubescence or otherwise the hairs of the leaves, and then it gets bulged and then it will break. Then we thought it must be all the salts must be coming out. But to our surprise we found it is only the sodium chloride which was coming out. And we got the X-ray diffraction done of the crystal and found it was nothing but sodium chloride. And when we were doing this work, some people from Gujarat fertilizers, they visited us. They said, when the MD was Mr. Vittal, he came and then he said, what is it you are doing? I said, we are growing plants with... I have one problem. Can you solve that problem? What is it? I asked. You see, I have Bait Island there, and that island nothing is growing. And the camels are brought right from Kutch, and they are left there for about 3 months. And we have to provide fodder for that. And government has said that it is not possible to provide fodder from the inland, so you have to do something. What I did is I planted Atriplex all through, and then it was used as a fodder for camel. Camel has got an excellent way of ruminating and then taking that whatever that it is there, and afterwards it will throw away the salt also. So there was no salt remaining in the body of the camel at all. It maintains the balance. So this was one thing which we did in Aliya Bait Island. Next, please. Then we thought, we have many plants which are growing on the sea coast, why not we see in what way we can use that? Then we found that Salicornia was one, but people were not knowing what for we should have this plant. So we collected this and then we extracted from this an oil which was about 30% oil which can be used as an edible oil. It has got some amount of resin, but that resin can also be separated and this can be used as a edible oil. We found that this plant is very wonderful plant. It takes the salt, stores the salts in different tissues. The major amount of sodium is found in the mesophyll region. It is not very much in the vascular or in the palisade region. In the palisade region it is so less, so the photosynthetic activity takes place and the plant is capable of producing whatever the food material that is required. Next please. Then when Dr. Naidamma visited, he told us, you see, your scientists simply spend money. You don't get money to the institute. I want every scientist must think about this. He said, you stop all your this seawater, this, that and all that. You concentrate on something else so that you can get some money. Then I thought, what can we do? When I was there in Arizona, I met one person called Miller. He was doing some work in jojoba. I said, give me some few seeds. I brought those seeds and started this jojoba work here. You know, this seed contains nearly 50% of oil and it is a replacement to sperm oil. So, we started growing this on the coastal sand dunes and many people took a lot of interest in this. Then we got quite a number of network projects and all that. And I thought it is better to have one more plant. Next please. And this is a plantation which we put up on the sea coast near Janjmir and selected quite a number of high yielding varieties and also those which could give high amount of oil. Next please. And we had some projects with other people, that is Chotila, we had a person who could invest some money and he wanted to have a jojoba plantation. So we helped him to take up the project. Similarly, we had, next please. You can see here, there is a lot of difference in the seed size and all that. Why I am telling all these things is because these are all plants which are not domesticated just like wheat, rice or other plants. These are all undomesticated plants. Even algal plants are also not domesticated. We like to domesticate them. We like to find out how much yield it can give, what kind of composition will be of the oil that we get, and then how we can increase the yield by certain technological methods. And then produce a variety which can be used by a farmer so that he can get more money from the land where he grows these plants. Next please. That's okay. This is of course a comparison with the sperm oil. Next. Where is this one? Oh. Next plant is, we thought when I was there in Thailand, these people were using all kinds of oil to run the engine. After 1,000 hours, they have to down the engine, remove it, and then decarbonize it, and then again run it. Then I thought, why not we also start doing it? When I was going to, from Visakhapatnam to Berhampur, on the way one day night I saw some people were lighting lamps. Then I stopped the jeep and went and saw there. They were using some kind of oil. It was so bright. I thought, what is this oil? It is so bright. It is not kerosene. But these fellows are using such a good oil, which is so good, and gives a very bright light. Then I asked them, can you give me some seeds? They gave me some seeds. I brought that, planted, and then extracted that oil. After extracting it, when we wanted to use it, we found that it cannot be used as such for the engine because it is the same Jatropha carcass which cannot be used as such for running the engines. Then we did the esterification and removed the triglyceride from that After that, we were able to use it straight away as a diesel for running this thing. But I could not do further, mainly because the cost involved in growing this plant and extraction is slightly higher than the diesel that is available today in the market. So unless the yield increases by a certain fold, we will not be able to make this a profitable venture. So I just kept it like that. Very interestingly, the work was carried out further in the institute, and they have taken patent and produced nearly 1,000 liters of diesel and supplied to many organizations, and also run our own vehicles with the biodiesel. This is a very important project for the nation. Why I am telling this is, we had a meeting with Dr. Swaminathan as the chairman on how to use the wasteland, how to increase the productivity of wasteland. Then many people, the forest department people suggested we could put eucalyptus, this, that and all that. They gave all kinds of figures. He was not satisfied. He said, no, I want something else to come, so that the person who wants to grow anything on the wasteland must be able to get something which will be a sustainable one. So we suggested that this will be the future of wasteland development project. Next, please. Well, all these things have been possible because we had many people who sponsored the projects for us, for both jojoba, Violi, and also, Violi I forgot to tell you. This is a plant which I introduced from Mexico. It contains rubber to the extent of about 10%, and it can be stored. The rubber will remain as such in the plant for any number of years. It doesn't get oxidized. Whereas in case of Hiva brasiliensis, once you tap it, you will have to process it. That's not the case with this. Can you put next? This is the plant. Next one, please. No. Next. Right. And what happened was, this rubber which we get is almost the same isoprene molecules we have as that of Hiva brasiliensis. But this is also an undomesticated plant. We have to domesticate this. So we made a lot of work on the yield, composition, and also the characteristics of the plants. Because these are all wild plants. They are not domesticated plants. And then we introduced a plant called as Simaruba glauca, which is a tree. It's a very interesting plant. This plant yields something like our almond nut. You break it, then you get kernel in that, very soft kernel. You squeeze it, you get about 50% of oil, which is a non-fat oil, which is a very good thing for people who have some problems. They can use this oil as for them. We had a project with one person in Kutch, and this plantation is existing in Kutch, in the Nalia region. He has been supplying some oil to some hotels there in Bombay. It's a low-fat butter oil. Next, please. This is the picture of Professor Boyko and Mrs. Boyko, who visited this institute in the year 1964. And we were doing the work with the tobacco. They were interested how you had chosen tobacco. I said, tobacco is a guinea pig. In all plant work, we first use tobacco as a guinea pig to find out whether it can tolerate or it can give better yield and all that. When we worked on it, we found that yield will not get reduced very much because it's only the leaf which we use. The nicotine content remained the same. But the only thing that was there was the burning capacity of the tobacco, which is cigarette tobacco, got reduced because of the chloride content. But we can just use that straight away for insecticidal purposes. And this is the Chief Justice of Gujarat High Court. He came here to see the institute. At that time he took interest on our sulfurization of jojoba oil, which we were using it for as a lubricant, we can use it. So he was very much interested in that. And Morarji Bhai Desai, the former Prime Minister, when he visited this institute, he was moving around. He asked me, what is that portion there you have put some props there? I said, sir, it's an old building. We have put some wooden structures there so that it doesn't fall. Who is sitting there? I am sitting there. I sit there, sir. I said, it is my laboratory. Gentlemen, do you know, I was imprisoned here for 6 months. So you are using my room as a laboratory. It's a very good thing you have done. And then he went on, when I explained to him about this rubber project, he took a lot of interest and he said, how much money you want for this rubber project? If you want about 20, 30 crores, 30 lakhs, I can give you. Start the project. I will ask the rubber industries to fund this. Unfortunately, next election he was not there, and then the whole project. So, gentlemen, this is how the genesis of this. From a small scheme, we became a section. From a section, we became a division. And then later on, we could make an impact. And this has now gone into the annals of the biosalinity work in the whole world. There are about 5 or 6 books which refer our work. And this is considered to be one of the schools in India which does active research on biosalinity. Thank you all very much.
I don't want to say anything. I have said so many things before. Please help me to tell him. Sir, it was a great moment for us